Hello everyone, my name is Pallavi Doka, and today I'll be explaining about the rubella virus and its microbiological aspects on how it affects the body as a viral infection, and why funding for vaccination is extremely crucial in protecting our society and future generations. So before I get started, I would like to note that rubella is not the same as measles or rubiola, as there are some slight differences between the two. Rubella is actually caused by a different virulent than measles and is slightly less severe as well. Another term for rubella would be German measles. However, both do have extremely similar noticeable symptoms, such as the erythematous rashes, eye redness, and mild to high fever. The rubella virus is part of the arbovirus class of diseases, which is specifically in the Togaviridae family, noted as the rubevirus. However, although it is part of the arbovirus class, it is the one virus that is not transmitted by an arthropod, so it becomes an exception, basically. Its only known reservoirs are humans and cannot be transmitted from plants or animals. The virus has a spherical shape to it, and the structure of the virus is shown here with the positive sense single-stranded RNA, or ssRNA, and it has roughly 9,757 nucleotides inside this icosahedral nucleocapsid. And right outside, covering your nucleocapsid, you have your lipid bilayer membrane envelope, which E1 and E2 glycoproteins are spiking out from it. And these glycoproteins have hemagglutinin in them, and they're used to bind onto receptors in the nasopharyngeal lymphoid tissues and regional lymph nodes, where their lytic cycle replication begins. The hemagglutinin is also responsible for binding the virus to the host cell so that the viral RNA genome, this squiggly thing, can be injected into the cell cytoplasm efficiently. So here's a more basic structural view of the virus, and here's a more magnetized view of it. And a fun fact is that the virus has the highest percentage of guanine to cytosine base pairs than any RNA virus out there, with roughly 70% of its genome being composed of them. The glycoproteins themselves are about 5 to 6 nanometers in length, but the entire virus is about 40 to 80 nanometers. So as I mentioned before in the previous slide, the virus has these protruding glycoprotein spikes, which are basically E1 and E1 proteins. So in general, all viruses use these glycoprotein spikes to attach and bind onto the cellular receptors that they are specific to in the body. The receptors recognize these viral molecules and make it easier for the virus to invade that area, since they're structured in a way to infect areas that are most easily accessible, basically to be the most efficient at doing their job. The glycoproteins make it easier to slip into the membrane of the cell they're trying to infect by attaching and fusing into it and multiplying in the cytoplasm. Rubella has an airborne mode of transmission, meaning it's passed on from one infected person to others through respiratory droplets from the virus. In other words, when one person might cough, sneeze, or talk, their mucus droplets and saliva transfer over to one person who's nearby, who's not careful enough, and could possibly end up in their nasal tract or mouth area. Most commonly, the nasopharyngeal tract, upper respiratory tract, and the regional lymph nodes are affected first and become the site of the virus's replication. The glycoproteins will attach themselves to the mucosal cells that line the epithelium, or inner part, of our respiratory system tracts and then will begin to multiply. Again, the glycoproteins will be specific to the receptors on these mucosal cells, hence why plants or animals can't necessarily contract rubella, since their mucosal cells have different receptors than ours. The virus is also called teratogenic, since a mother who has contracted rubella in her first trimester could pass it on to her fetus, and this causes developmental malformations in her embryo, which I'll explain about later in the coming slides. The virus can enter the placenta and infect the fetus in the uterus. At that point, the baby has what's called congenital rubella syndrome. The virus replicates in the lymph nodes and the nasopharynx, which is the tube connecting the nasal cavity, and the soft palate. Between 5 to 7 days after the infection, the virus will have spread to the rest of the body through the bloodstream, or hematogenously during viremia. It's not yet completely clear what exactly the cellular receptor for rubella is yet. Some say it's the P32 protein, and some say it's the MOG protein, or the myelin oligodendrocyte, but it's expressed in the CNS. So naturally, people will have theories that the virus has other receptors that are responsible for allowing the entry of it into susceptible cells. So here's a picture of the airborne transmission, and this is the entry and exit portal, and these are some symptoms that result. And here is the congenital rubella syndrome depiction being shown with this arrow. And it also has heart defects, which I'll explain later. So the rubella infection causes direct damage to the host cells. As we learned, the lytic cycle results in the death of the host cell and reproduces new phages, called the virulent phages, which lies or break out of the host cell's wall, releasing the viruses. The infected cells produce IFN alpha and beta, which are innate cytokine responses that are important mediators in detecting when the body is trying to defend against a viral infection. They alert the organism in case of a viral infection by the detection of abnormal double-stranded DNA, and also to inhibit the virus multiplication by acting on the translation of infected cells. So basically, when that RNA genome gets, placed into the cell, that's when the cytokine responses will start to go off and alert the cell that a virus has been injected. So these infected cells produce these responses, which inhibits viral replication, cell proliferation, and increases lytic potential of natural killer cells. The infected cells are then killed off by cytolysis. In simpler terms, basically in this diagram, the virus first attaches to the cell surface and then enters the cell. The virus takes over the cell and forces it to make viruses by injecting its genomic RNA. Once the viruses are fully ready, they break out of the cell, killing it, and then repeat the process over and over again, joining other cells. The virus is identified using nasopharyngeal throat swabs taken 6 days before or after the appearance of those rashes. Using the cell cultured in shell vials, antigens can be detected by using immunofluorescence methods, kind of like what we did in the pea-glow jellyfish lab. The presence of IgM rubella antibodies in the blood indicates a recent infection, and the presence of IgG antibodies indicates a past rubella infection or that a vaccine has been given and is giving adequate protection. So in other words, if you have a high count of IgM rubella antibodies, it could be because you've been recently infected. But if you have a high count of IgG or immunoglobulin count, this could possibly mean that you've been administered a vaccine or that you've already had the virus in the past and you are now immune to it. They are administered subcutaneously through shots and are usually given right after birth at the age of 1 through 12, both tests. IgG stays in your bloodstream for life, so it's very unlikely to even get rubella once, let alone twice. One dose of the MMR, or mumps, measles, rubella vaccine, is 97% effective at prevention. The vaccine is given in 2 doses, as I mentioned before, one at the age of between 12 to 15 months of age, and the second one at 4 to 6 years. The virus is extremely important to get since it's a highly contagious virus. There is no direct treatment per se, and protection acquired at only one point of time provides lifelong protection and future generations to be safe. Pregnant women don't have to worry about their child contracting the effects of CRS, such as blindness, mental retardation, and the other effects on the central nervous system. So now a little bit about how the vaccine actually works. The rubella vaccine is a live, weakened form of natural rubella virus. The rubella vaccine is weakened by a process called cell culture adaptation, which modifies natural rubella so that it behaves very differently once it's injected into the body. They normally grow in cells that line in the back of the throat anyways, so natural rubella virus reproduces itself thousands of times, occasionally causes severe disease, and is passed on to the next person unchanged. However, cell culture adaptation changes all of that. Natural rubella virus was first taken from someone infected with rubella. The virus was then grown in human embryo fibroblast cells. These cells were first obtained from an elective termination of one pregnancy in England in the early 1960s. These same embryonic cells have continued to grow in the laboratory and are used to make rubella vaccines today. Fibroblast cells are the cells needed to hold skin and other connective tissue together. By growing rubella viruses in human embryo fibroblast cells, it became less and less able to grow in human cells that lined the back of the throat or in cells of an unborn child. This happened because the genes that told rubella virus how to reproduce itself were changed. Now the virus reproduced itself very poorly. When this vaccine virus, a now modified form of the natural virus, was put back into other children, it grew very poorly. Whereas natural rubella virus reproduced itself thousands of times during natural infection, the rubella virus vaccine reproduced itself probably fewer than 20 times. That is why natural rubella viruses cause illness, but the rubella vaccine does not. This vaccine should definitely continue to be funded just as it is right now. As I touched on before, rubella can cause miscarriages and serious birth defects to a fetus if a woman is infected while pregnant. This epidemiology bar graph shows the history behind the numbers of countries who have recognized the importance of the MMR vaccine in the growing years. So we can see here that so many countries have adopted the use of the vaccine because it's provided so much protection to the people, especially to the females. And here we see this nice steady incline of the global population living with the vaccine. We should obviously continue to be funded to administer the vaccine because the last thing we would want is for our babies to not be able to live up to their healthiest potential, and we naturally want to give them the best quality of life. And once you get the MMR vaccine, it also protects against mumps, measles, and varicella as well. And lastly, this is Bella Baby. It's a cute and bit gloomy way to remember what the symptoms of the congenital rubella are. So first we have the head, which is significantly smaller than the body, which represents microcephaly. You can see that there are C's for her eyes, which represents cataracts. And here she has some earrings, which are in the letters D, to represent deafness. And here she has a hole in her heart, which depicts PDA, or patent ductus arteriosus. By the way, her heart got broken at the toga party she was at, hence why she's wearing one. And because rubella is an enveloped virus, which is part of the toga virus family, a way to remember it. And I gave her a big E for her hair to stand for encephalitis, or inflammation of the brain. And then finally, she has some cute blush here to represent the blueberry muffin rashes that result from the viral infection. I hope this video helped you learn a little bit about rubella and why vaccination is important, so be sure to keep track of them and be aware of your surroundings at all times. Thank you.